Recording is now live. Thank you everyone for coming to April's Women in Aerospace Talk. We are so pleased to welcome Egle, who goes by that. She is the one and only Egle at NASA. So that is how she prefers to be called. My name is Shana Gifford. Dr. Gifford, pronouns are she, her. And I am very pleased to have you here today. I know we have a few people who are here for the first time, so I'll walk you through Women in Aerospace Medicine, our mission, our vision, our values. We are a diversity initiative of the Aerospace Medicine Student and Resident Organization, the international organization for people who aspire to aerospace medicine research or medical practice. We came into being in order to create opportunities for leadership, support, and community specifically for female identifying people and minorities, but all are welcome. This is a community for everyone. All of the speakers that we've had over the last 2 years, and you'll see a picture of them shortly, have had to work very hard to get where we want to be. And we want the same success for you, ideally without the struggle. So that's our mission, to advocate for environmental, space, and aviation medicine success among students, residents, and aspiring researchers and practitioners. This is our board, and 2 years ago, it was me and Dr. Melissa Jordan, a member of the US Navy in the upper left-hand corner, just me and Melly, and then along came everybody else. That's Dr. Laurel Kane. She is an MD, PhD, who is currently at Oxford studying astrophysics. This is Dr. Shilpi Ganguly from the University of Miami. That is Dr. Caitlin McIntosh. She works for Lockheed Martin. That is Dr. Eleanor Frost, also known as the space surgeon, whom some of you may know, and that's Ksenia, who is the former head of AMSRO. This is our board of aerospace, Women in Aerospace Medicine. In just a few weeks, Women in Aerospace Medicine is kind of doing a small takeover of ASMA. We're going to have a lot of speakers. And if you will be in attendance at the annual Aerospace Medicine Conference in Reno, please come here. Victoria, Riley, Tanya, Nina, Lynn, Eleanor, Lauren, and Shauna, and myself. We're all going to be talking. We're basically taking the place over. Everyone, please mic your mics. All right, hang on 1 second. Thank you, Diego, appreciate it. So, okay, here we are. Let me just back up for 1 second here. Good, more good news. Victoria Tucci, who will be speaking at ASMA, won an incredible scholarship in memory of Anita Montree. So we're very, very proud of Victoria, who's been winning like a scholarship every other week this year. She's really been killing it. More good news. Mission Astro Access is back. So for those of you who know anything about it, this is a disability initiative so that people who are what I like to say adapted, either in mobility or vision, are able to attain the same access to space as everybody else. And in many ways, they are better suited for the space environment. Many of them don't walk with their legs. They propel with wheelchairs or by other means. It wouldn't occur to them to try to use their legs to get through space. Those people who have vision and hearing issues, they're very, very good at adapting to their environments. Space is for everybody, and applications are open for our next mission to microgravity, which will be in December 2020. Please encourage anyone you know who might be eligible to apply. Axiom 1 is back. There's flight surgeon Natasha Shaw. We're very proud that this first mission went off so flawlessly. Congratulations, Axiom. We're also very excited that, fingers crossed, pretty soon we're going to have the first mission ever for Artemis, which is going to use an orbit that no one has ever flown before, as well as a medical monitoring dummy mannequin, space representative, not a real person, but somebody who is going to fly along and show us what happens to the cabin environment. We are all very excited about that. These are our former speakers. We've had some incredible speakers over the last 2 years: astronauts, space doctors, members of the military, people who run entire branches of NASA and entire academic departments. And we're very pleased that we've had them and very honored. There are a few more patches available, just a few. So if you'd like a Women in Aerospace Medicine patch, feel free to let me know and I can send you a link to order it. Without any further ado, Egle. Egle prefers to go by just that. And she is actually the first speaker we've had who I have not met in person. So I'm very grateful that she came today, because I started my career in neuroscience, and neuroscience is fundamental to our life experience here on space and everywhere. Egle works on something very hard, which is modeling human organs in space. This is what we call a wicked problem. And a wicked problem, some examples of wicked problems we deal with every day in space are fluid dynamics, circulation, sleep, immune function, which is also part of Egle's research, metabolism, and space radiation. How do you model this on Earth? This is a wicked problem. Egle works on that and also serves as the course director for the NASA STAR program, which is something that everyone here might be able to avail themselves of. So without further ado, I'd like to pass off the entire presentation to Egle so that she can take it away and tell us more about all of this.
Great, thank you very much for this wonderful introduction. And let's see if the screen sharing still works.
You may need to go, yes, full screen and you'll be all set.
Is it working okay?
So
do you see the correct display? Excellent.
We do.
Okay, so thank you very much for well, waking up or not going to bed on this fine Saturday to listen to me talk about what I happen to be studying in the radiation biophysics lab at NASA Ames, which is the effects of deep space radiation on the nervous system and the immune system. And I'm a research scientist and principal investigator there in the Space Biosciences division. So everything I say today is my words and not those of the government. So first, a little bit about me and my journey. So as you may guess from my rather complicated name and my accent, I was probably not born in the US. I, in fact, was born and raised in Vilnius, Lithuania. And I've been dreaming about NASA and about human space exploration since I was a preteen. And it only took me a bit over 25 years to get here. I went through a bit of a circuitous route. I could not find any way to study space biology as an undergrad. So instead, I went for neuroscience because brain seemed to be just as little explored as responses to spaceflight. And I came to the US with $500 in my pocket on full financial aid. And I realized I needed a job the first week at Harvard when I saw how much the textbooks were costing here. So I studied neuroscience and I continue to study neuroscience for my PhD and continue to study the interaction of the gut microbiome and immune system still in neurological disease and multiple sclerosis during my postdoc. And now I am studying the neurological and neuroimmune responses to spaceflight, which are in fact rather similar to studying them in response to a disease, except that this time my disease or my environmental perturbation is called space. So what causes the main health risks of space exploration? Well, first of all, it is changes in gravity, microgravity in flight, partial gravity on the moon and on Mars, and hypergravity or increased gravity during launch and landing. Also, confinement and isolation, which we are all very familiar with after the last 2 years. Distance from Earth, which obviously impedes medical care. And what's called hostile and close environment, which is kind of an umbrella term that encompasses high carbon dioxide, ambient noise, circadian rhythm and sleep disruption, and limited environmental microbiome, which does have an effect on all human organ systems too. And of course, primarily what I happen to study is the exposure to space radiation. So the health hazards associated with these environmental factors are numerous. And specifically, radiation damages DNA and leads to mutations, which are carcinogenic. At the cellular level, it causes oxidative stress, which leads to tissue and organ degeneration across multiple tissues and organs and multiple organ systems. Now, the nervous system is one of the least studied. That's why it's not even listed in this paper that came out less than 2 years ago, because there are not very many studies on the space radiation in the nervous system. However, it is generally a degenerative and inflammatory and oxidative stress causing factor. And we particularly want to study space radiation because we are planning for lunar and Mars missions. Now, on Earth, we are protected from galactocosmic radiation by the magnetic field of the Earth. The International Space Station is close enough that it still falls within that protective magnetic field, and also astronauts, for the most part, do not go on such prolonged missions as especially Mars missions will have to be. Of course, going towards the moon and to Mars, we will have to leave the protective magnetic field of the Earth and be exposed to primarily what is called the galactic cosmic rays, which is the deep space radiation. Galactic cosmic rays is a bit of a misnomer because it's actually a shower of particles. These particles originate in supernovas and they permeate deep space. They are very hard to shield from, and they are primarily protons, so hydrogen nuclei, some helium nuclei, and the rest of them are, if you imagine a periodic table, all the way through iron. Iron is a particularly biologically effective and biologically damaging, we must say, component of galactic cosmic radiation. Now, the doses are not terribly more different on the way to the moon and to Mars, especially on lunar and Martian surface, than they are on the ISS. But they are heavier in the particle, in the high mass, high energy particle component. And also, of course, they all sum up during the prolonged mission. Since I can't send my samples to the moon, expose them to deep space radiation there, instead I imitate deep space radiation on ground, specifically using particle accelerators at a NASA Space Radiation Lab, which is located in the Brookhaven National Lab. And here you see me at the beamline, where I just put in my samples. Here you see what it looks like when you look straight into the proton source. Of course, the Star Trek uniform is entirely optional. Now, specifically, I am interested in the effects of deep space radiation on the nervous system, as I mentioned. We know a little bit about them from exposing model organisms, primarily mice and rats, to simulated galactic cosmic rays or their components, right? All these particles, especially iron particles. They lead to behavioral deficits, including memory and social behavior, that are accompanied by neuroinflammation and neurodegeneration at the cellular and tissue level. But radiation doesn't just affect the brain by itself, it also affects the rest of the body. And the brain and the body interact through the blood-brain barrier. From terrestrial diseases, we know that if the blood-brain barrier is compromised, then the systemic whole body responses, whole body damage can exacerbate inflammation in the brain, which is particularly important to us because neuroinflammation is already one of the outcomes of space radiation. So we are very interested in the effects of space radiation on the blood-brain barrier, which have been studied very little, especially in human models. Now, this is another part of what I do that has been particularly fun for me, which is building organ models, which I believe are a fantastic approach for studying the biological effects of space radiation and other deep space stressors. So how do we build an organ model? We start with primary cells that can be isolated from human donors or model organisms, and then they're converted to induced pluripotent stem cells, which then get differentiated into cells of an organ of interest, in my case, the brain. Then those cells can be used for either creating organoids or seeding organ-on-a-chip microphysiological systems. Then those systems can be exposed to simulated spaceflight stressors, which is mostly what I do, or even flown as payloads. We've had a few organ model payloads already, and hopefully we'll have many more in the future. And together, the information that they provide tell us more about the fundamental biological responses to spaceflight, also targets for biomarker and countermeasure development, and allow us to compare flight with terrestrial diseases for the mutual benefit of both space and terrestrial biomedicine. Also, very notably, if you want to eventually turn these models into payloads, they high throughput, have a small footprint, can be automated without the need for crew time, and can be developed from multiple organisms, both multiple types of organisms, such as human and model organisms, and multiple individuals to address the variability. For example, the variability of human responses to space radiation. Now, as I mentioned, I am interested in the blood-brain barrier, so I want to model blood-brain barrier. What blood-brain barrier is, it is a tube that is a blood vessel, right? Its walls are formed of endothelial cells that are connected with each other through tight junctions. There's brain tissue outside. Where particularly important cells for us are astrocytes because they can regulate barrier permeability, but also multiple aspects of neuronal health and neuroinflammation. So I built a very simple, high throughput blood-brain barrier model using a commercially available system. This is a Mimetas organoplate system. It's basically a standard 384 well plate, which is very standard for those who study, who do a lot of biological lab research. And it is a collection of chips, up to 96 chips per plate, where each chip is composed of 2 channels or 3 channels. Here you see 2-channel chip for as an example. And this is what we seeded the chip with, which are different types of cells. So we have astrocytes at the bottom, or no astrocytes to address the role, and endothelial cells forming a 3D tube in the top channel. Again, very simplified for very basic questions, but human and high throughput. And very nicely, our endothelial cells formed a 3D tube, as you can see here, of a blood vessel. And here I also stained for astrocytes as a cytoplasmic marker to see the green astrocytes with the teal endothelial cells. This is another example of a 3-lane chip to see that we can also grow astrocytes in the middle and have a tube at the top and basically another tube at the bottom. The main manuscript for this is currently under review. Okay, so we built this model. Next, we of course wanted to expose it to simulated deep space radiation. And we did 2 types. We chose iron particles because they are particularly biologically effective component of galactic cosmic rays, and a simulation of multiple components. So protons, helium ions, and different types of other particles. And so what happens? First, by measuring the leakiness of the blood-brain barrier, so how much it is open, because remember, when it's open, it exacerbates inflammation and it's basically bad and harmful for the brain. The leakiness we can measure by taking a dye and loading it into the vascular channel. And if a dye is fluorescent and the barrier is leaky, the fluorescent dye is going to leak to the neighboring channel and increase the fluorescence in the neighboring channel. So if you quantify the relative fluorescence in the neighboring channel over the vascular channel, it will be closer to 1. If the barrier is perfectly closed, well, then nothing leaks out and the relative fluorescence will be closer to 0. In addition to leakiness, we looked at the morphology, at different cell types, at the cell shapes, at different proteins they express, markers of oxidative stress and inflammation. And basically for all of this, we asked 2 main questions. The first one is, what radiation do to blood-brain barrier? The second is, what do astrocytes do? Do they make it worse or do they make it better, or does it change over time? So early after radiation, about 24 hours, the main thing that radiation did was a dose-dependent increase in vascular permeability. It made blood vessels leaky, and astrocytes made them even leakier. So here we see a representative graph of leakiness of blood vessels. Again, the closer you are to 1, the leakier the vessel. Endothelial cells alone are without shading, and endothelial cells and astrocytes are with shading. Blue is no irradiation. Increased shades of red are increased doses of radiation. Basically, you can see that radiation makes them leaky and astrocytes make them even leakier, which is not necessarily a beneficial role, but can also be helpful to some degree early, because by opening blood-brain barrier, you would let immune cells in the brain that would be able to clear the debris and the dead cells. So early on, leakiness sort of has a twofold role. What primarily happens later? Astrocytes form a scar. They start expressing a protein called GFAP, or glial fibrillary acidic protein, and basically behave as if you had a brain injury or a stroke, which I incidentally studied in my PhD. By 3 and 7 days after irradiation, the more radiation you have, the more scarring of astrocytes you will get. That is a more protective phenotype. In addition, when we measured oxidative stress, here we see again multiple time points after radiation, after multiple types of irradiation, both iron and the whole simulated galactic cosmic rays. We saw that first of all, radiation increases oxidative stress in our model. Again, blue is unirradiated, increased shades of red are increased doses of radiation, and astrocytes limited. So radiation is increasing oxidative stress, astrocytes make it increase it less. Still increase, but less. In this way, having a more protective role, especially later after radiation. And we did very similar measurements with various inflammatory molecules, cytokines and chemokines, which basically signal to the body that there is inflammation going on in the brain and we need to attract immune cells into the brain. Radiation increases them and astrocytes limit it. Which again is more beneficial. And that is something very similar to what astrocytes do in response to a brain injury or stroke. And it's even similar in terms of days, as in within the first 24 hours, they are more opening the blood brain barrier, more pro inflammatory. But by the time it's a week after the injury, they become more anti inflammatory and more helpful in reducing damage made by the insult, in this case, radiation. Okay, so in summary, in this part that I told you is we have developed a simplified high throughput human neurovascular model. We exposed it to simulated galactic cosmic rays or their components, iron particles, and we found out that radiation makes the vessels leaky, and it increases oxidative stress and it increases inflammation for at least a week. In addition, astrocytes have mixed effects. They reduce oxidative stress, which is good, and reduce inflammation, which is good, but earlier makes the vessels leakier, which can be harmful or beneficial sort of together. So this kind of activity and this kind of role and the switch to this protective phenotype makes a very interesting candidate for developing countermeasures or therapeutics that would specifically target astrocytes to further reduce the oxidative stress and inflammation caused by radiation. So this is something that we've been building in the lab. But we also have multiple other research directions in which we are actively working on. And I would like to give you an overview of what other aspects of neuroimmune responses to space radiation we are studying. So the very first one is basically looking at the variability of radiosensitivity. Because, as I mentioned, you can develop induced pluripotent stem cells from a variety of subjects and use them all to turn them into cells of interest and then develop organoid or organ on a chip models, again, of interest that are personalized. And then use them basically for example, to address the individual responses to radiation, individual risks, and develop personalized therapeutics. And also compare them to address the factors that distinguish the worst responders from the best responders, which would give more information about the mechanisms that control radiosensitivity. However, well, how variable are people? Because if everyone's cells respond to radiation in the exact same way, then this personalized approach and variability studies are not particularly interesting anymore. So we address the variability of human radiosensitivity by isolating immune cells from the blood of over 700 healthy subjects and exposing them to different doses and types of radiation. Iron ions, which I keep mentioning, which are sort of the key biologically active component of galactic cosmic rays. Two other components of galactic cosmic rays, which are argon and silicon particles, and also gamma rays as controls. And then we studied what happened to those cells. at baseline before exposure as a control, and at multiple time points after exposure. We looked at oxidative stress, at DNA damage and repair, at various secreted factors, and so on. And we also sequence these cells, so we sequence these people to understand, well, if there is a variability in radiation responses, then which genes and which other factors are they associated with? And those associations will give us more information about the potential biomarkers. Can we tell that somebody is more radiosensitive than others? And also about targets for developing countermeasures. So if we know which genes and which proteins are associated with making people's cells more radio resistant, can we then develop pharmaceutical that would target those particular proteins and sort of improve the radio resistance from the sensitive level to the resistant level. And somewhat fortunately for the study, we did see the incredible variation in people's cells radio responses to radiation, radio sensitivity. So here I'm just showing one representative graph of basically looking at DNA damage marker. DNA damage marker is a protein 53BP1. These are not human cells, these are just representative cells showing nuclei here in blue and DNA damage after radiation here in green. You can see that particles were just going straight through this nucleus, given dot, 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 dot of DNA damage sites across the board. And you can count the sites and the average sites per nucleus and say, okay, given the same, if we give the same dose and the same type of radiation to multiple people, will they have the same amount of DNA damage on average? And they have hugely different amount. Here are different differences of people's DNA damage without getting any kind of radiation at zero. And then after getting increasing different doses and types of radiation. At 4 hours afterwards. So if on the y-axis you see the average DNA damage, and this is sort of normalized, you see a huge spread of some people getting multiple times as much damage as others. In addition, some people respond very little to low doses of radiation and need high doses for a response. These are the hyposensitive people. Others respond very strongly to even low doses of radiation and then sort of reach the ceiling. These are the hypersensitive people. So the variability is huge, and we have already identified some interesting genomic associations with it, and we are continuing to work with it. And by the way, the self-reported male-female do not change in DNA damage, but age does increase DNA damage at baseline without any irradiation, and reduces the ability to respond to radiation, which is very interesting finding as well, given that we are now considering people of very different ages potentially traveling to space. So we will keep you posted on the further work in the study, but it is already very promising and hopefully will give us a lot of targets for potential countermeasure development. We are also working hard on both the immune cells and especially neurovascular models to address another problem of simulating space radiation on Earth, which is dose rate. So generally, when you're going to Mars, you're going to receive whatever dose it is through the 3-year trip to Mars. Now, when you're simulating it on Earth, you're going to take your cells or your mouse and give the same dose to that biological sample over 30 minutes. Well, that's not a very good representation, but that's kind of the best we have. So instead, we are trying to at least build gamma rays, so they are slightly less biologically effective because they're photons, not particles. But we're building a model that would allow low dose rate chronic irradiation. So not all at once over a few minutes, but chronically, continuously at low dose rate. For that, we built an environmental chamber where we basically can put in a cobalt gamma ray source, as well as other simulators of other spaceflight factors, such as microgravity by using a clinostat, as well as high carbon dioxide by increasing carbon dioxide in the chamber, and so on. And we right now have two major studies, one on neurovascular model responses to a high dose rate versus low dose rate radiation, so sort of to giving acutely versus chronically, and another one on human immune cell responses to, again, acutely or chronically given radiation. And that is very important because that will inform us of a bit more of what changes when the rate of radiation becomes lower. When from an acute irradiation you are moving to continuous and chronic. Does it make the tissue responses better because there's enough time for repair, or does it make them worse because the response becomes like multiple ongoing repeated injuries, even though low level injuries, but ongoing and sort of cumulative? We don't really know. Nobody has looked at it in this kind of comparative fashion yet. There are some studies in mice that are worth looking at, and they do seem that the responses to low dose rate are still quite strong. Also, of course, what I presented to you was a very simplified basic model system of just two main cell types, and they're great because they are high throughput, because they grow fast, and allow multiple measurements quite quickly. But ideally, we do want to develop more complex organ systems and connect multiple organs into a single circulatory system. So we recently, together with the two collaborators listed below from Rensselaer and Johns Hopkins University School of Medicine, we received NASA funding to build long-lasting model systems of multiple organ models at once. So I'm going to work on the brain, Dr. Blaber will work on the liver, and Dr. Travakar will work on the gut. In addition, what we will be building is an automated connection for nutrient management, for waste management, and for sampling the system to address the health of the system and basically the functional outcomes of this irradiation. And the automation is very important if we are ever planning to turn this into a payload to reduce crew time. So we have a hypothesis that the effects of space radiation on the system will be similar to aging, because there are many biological outcomes of radiation, especially, say, on the brain, including oxidative stress, including inflammation, including blood-brain barrier permeability, which I have just showed, that also to some degree occurred during aging. So that is another interesting research direction, which I think is particularly important in terms of considering how we're going to turn our ground models into spaceflight suitable models. And we are, like I mentioned, these models allow us to compare terrestrial and space diseases. So we are working with University of California, San Francisco to repurpose terrestrial therapeutics for spaceflight. There is a giant database of databases called SPOKE, which contains all sorts of different biomedical information, including about compounds, including about their therapeutic effects, but also their side effects and various diseases and symptoms and gene expression associated with it. So we took the database already and fed in what we know about gene expression in spaceflight from mouse models and said, okay, which diseases, which new things come up? Which diseases and which symptoms do they all resemble? And we got a lot of them, including a very new one, which is space motion sickness, which is quite a nice example, if you can imagine, because in mice, obviously your mouse cannot tell you that it is motion sick. However, if you just look at the gene expression in the mouse's immune cells and then compare it to what's known in gene expression in humans that they're suffering from various conditions that include motion sickness or nausea, that actually turns out to be quite similar. So we can start inferring things that we wouldn't be able to infer otherwise. And we are continuing this work as well. Now, very importantly, in addition to that, we of course are interested in the responses to true spaceflight, for which we are studying mouse models and gene expression changes in spaceflight mouse brains and immune cells and also hearts. Now, this study I specifically want to mention to you because it is something that a lot of you can do as well. When we were interested in, okay, we're finding all these changes in blood-brain barrier, in inflammation, in oxidative stress in our model, what happens in mice? Well, I haven't flown any mice yet, but I can go to the Biological Scientific Collection, NBISC at NASA, and request samples for free, as long as they have a good write a good scientific reasoning for it. And then you can use them to study whatever you want, in this case, changes in gene expression. In addition, after we did all the sequencing, which I can imagine, not everyone can do that because not everyone can afford to do the sequencing, and not also, I think it may be limited to US persons. So when we did all the gene sequencing, we put it all on NASA Gene Lab, which is an open access repository of all space omics data. So all types of gene expression and other data. And it's there now. And while we are studying this in our lab, any of you can go to NASA Gene Lab and access the same data and study the same data and publish on the same data. And this is sort of going towards the end. That's something I particularly wanted to emphasize is the Gene Lab and the open science initiatives that have analysis working group for professional scientists, as well as more teaching initiatives for universities and even high school students. And I would strongly recommend you check them out or email me and I'll tell you more about them. If you are more of a physicist, you may want to look at the equivalent of Gene Lab just for physical sciences. And if you want to get samples or other types of data, that is also available free of charge, open access, of course, with a scientific justification. And Gene Lab for high school and universities is only one of the many, many educational and outreach programs that are done at NASA and NASA Space Biology that I am somewhat involved in, quite a lot of them. There's a big one for undergrads every summer that is an internship program at Ames called Space Life Sciences Training Program. Postgraduates and others should definitely check out the Young Scientist program at one of our contractors at Blue Marble Space Institute of Science. There's also a postdoctoral program. And there is a Space Apps Challenge program that I would recommend all of you looking into and possibly even leading a group that would respond to a challenge. There will be 2 challenges from a space biology one, at least 2, and one of them will be led by me. So just keep your eyes open for early June. I can't tell you yet what the challenge will be, though it is ready, but we will post it all online on the Space Apps Challenge website. And of course, I am the course director of the NASA STAR program. Now, NASA STAR program is for principal investigators and senior researchers. And it is basically for people who have been doing some variety of biology adjacent research and really want to go into space biology, but don't know how. So it teaches both fundamental space biology and also more practical aspects of flight payloads or biological flight payloads. It is virtual, it is international. The only requirement is an advanced degree such as PhD or MD because it's sort of for that level investigators who may have their own missions. And the deadline for applications this year is first. So please apply, please spread the word. I especially would love to see more applications from traditionally underrepresented people and from traditionally underrepresented institutions. Right, and now thank you very much. This is my group at NASA Ames and all our collaborators, and Specialist Sylvan Costas is the other principal investigator and the lead principal investigator in our radiation biophysics lab, and of course, many collaborators and funding sources. And this is how happy I am at midnight when I get to irradiate my cells at the NASA Space Radiation Laboratory. And this is how you can reach me. my first name dot last name at nasa.gov, which is also on my official NASA website, which you can just find out if you Google me. So thank you very much. We will end the show, and we have 15 minutes for various questions. Let's see if I can stop sharing and actually see you folks. Great. Now it's the first time I actually get back to seeing you. So let's see.
So great to see you all. So great. Thank you so much. We have such incredible questions in the chat. I'm actually sad that we're going to stop recording because I think these are great questions, but people have the right to turn their cameras on and ask their questions in comfort and privacy. So I want to thank you, and I'm going to stop the recording now. Anybody out there listening on YouTube who wants to get in touch, please feel free to reach out to Egle and or Women in Aerospace Medicine. We'd be happy to put you in touch. All right, and with that, I'm going to